Hey everyone, and welcome. Today, we are going on a pretty wild microscopic journey. We're going to demystify one of the most talked about procedures of our time, the COVID-19 PCR test. We're going to follow a sample right from the swab all the way to that final result. So let's follow along and see where the science takes us. I mean, think about it. You get the swab, they put it in a tube, and then what? It just kind of disappears into a lab and later a result pops up. But the story of what happens in between? Well, it's actually this fascinating piece of high-tech detective work. So the first thing you need to know is that this isn't just a simple chemical they splash on a sample. No, the whole journey starts with a very specialized package. You can basically think of it as a detective's kit, and it's designed for one very, very specific mission. And what's so cool about this is that it's not just one thing. It's a complete, ready-to-go system. It uses what's called real-time PCR technology, and the kit has every single reagent and control that a scientist needs to hunt for the virus's genetic material with just incredible precision. Okay, so how does this detective kit actually find its target? Well, like any good detective, it has to know exactly what it's looking for. In this case, it's on the hunt for a very particular genetic signal. And that signal is the virus's RNA. The easiest way to think about RNA is like a unique instruction manual for the virus. It's different from our DNA, and more importantly, the RNA for COVID-19 is totally unique to that virus. It's the genetic fingerprint that this entire test is built to find. To find this specific RNA, the kit unleashes what I like to call genetic bloodhounds. Now, scientifically, they're called primers and probes, but these are tiny, custom-designed molecules that are engineered to be a perfect match. They will only seek out and grab onto the genetic sequence of the COVID-19 virus. Their focus is, well, absolute. So how do these bloodhounds tell us they found something? Well, they're carrying a microscopic glow stick, a fluorescent molecule. The probe that's hunting for the virus is tagged with a dye called FAM, and a separate control probe is tagged with a different dye called HEX. And these little glow sticks are designed to light up only when they find and lock on to their specific genetic target. All right, let's head over to the lab bench and see how this all comes together. This next phase is all about preparation, and it's kind of like following a very precise, very detailed recipe to make sure the test runs absolutely perfectly. It really boils down to this three-step process. First, the lab tech has to carefully extract all the RNA out of the patient's sample. Second, they prepare something called a master mix, which has those genetic bloodhounds and all the other ingredients they need. And finally, they combine that master mix with the RNA they got from the sample. Now everything is set for the main event. And here we are. This is the heart of the PCR test. All of that careful prep work now goes into a machine that acts like a super high-speed genetic photocopier. This is where the real magic of amplification happens. You see, here's the problem. Even if the virus is in the sample, there might only be a tiny, completely undetectable amount of its RNA. The PCR machine solves this by running through these cycles of heating and cooling, which causes the RNA to get copied over and over and over again, exponentially. And you just got to see the power of this process. After one cycle, you've got 2 copies. After 2, you have 4. After 3, 8. This particular test runs for 50 cycles. By the time it's done, if even one single strand of viral RNA was in there at the start, it has been amplified into billions of copies. Suddenly, what was undetectable becomes completely unmissable. So all of this work, the extraction, the mixing, the endless copying, it's all designed to lead to one simple thing, a clear, reliable answer. So how does the machine actually decode the final result? It all comes back to those glow sticks we were talking about. The machine is looking for light signals from 2 different channels. The FAM channel tells us if the virus is there. And that HEX channel is for an internal control, which basically just confirms the test itself worked the way it was supposed to. By looking at which signals light up, the machine can deliver a really clear result. So let's break it down. If the machine sees that FAM signal light up, it means the virus's RNA was found and copied. That's a positive result. If it only sees the control HEX signal, that means the test ran perfectly, but no virus RNA was found. That's your negative. And if neither of them lights up, well, that means something went wrong somewhere and the test is invalid. So how confident can we be in these results? I mean, could the bloodhounds get confused? Well, the data is really impressive. To make sure it was accurate, the test was challenged with a whole bunch of other viruses, including other coronaviruses like SARS and MERS. It correctly ignored all of them, which just proves that those genetic bloodhounds are incredibly specific to their one and only target. So, from a simple swab to a complex chain of molecular reactions with genetic bloodhounds, tiny glow sticks, and a high-speed photocopier, it is an absolutely incredible journey. It's a great reminder that behind every simple positive or negative result, there's a whole world of powerful and precise science at work.